Yes, very good evening. So today we are here to discuss the questions of nerve muscle physiology. So myself Dr. Kamal Jindal, and I would be starting with the first question now, right? I hope the audio and video is fine. Yes, just let me know. Yes, very good evening. So today we are here to discuss the questions of nerve muscle physiology. So myself Dr. Kamal Jindal, and I will be starting with the first question now, right? I hope the audio and video is fine. Yes, just let me know. Yes, so let's begin with the first question. In cardiac muscle action potential, phase 3 depicts which of one of the following phases, right? So what is phase 3 in cardiac muscle? Remember, when we talk about the cardiac muscle plateau potential, is I am asking you this question, then this is the figure of the plateau potential, right? In this there are various phases. This is phase 0, which is rapid depolarization. Then we have early or initial repolarization, which is phase 1, right? This is known as early or initial repolarization. Early or initial repolarization. Then this is the phase 2, which is known as the plateau phase, and this is the phase 3, which is known as final or late repolarization. Late or final repolarization. Some people also call it delayed, right? Late or final or delayed repolarization. So prepotential is not in the cardiac muscle action potential. Prepotential will be seen in the SA node, right? Pacemaker potential. So the answer was phase 3 is late repolarization, right? I hope you got it correct. Right? So fine, let's go for the second question. Question number 2 was, which of the following is responsible for elasticity in skeletal muscle? Remember, it's an important question. The answer is titin or titin. The titin is the largest protein which is responsible for the muscle elasticity. Titin is responsible for, it is the largest protein responsible for the muscle elasticity, right? It is the largest protein responsible for muscle elasticity, right? Titin, remember, is responsible for the muscle elasticity. It is the largest protein. It joins M line to the Z line, right? M line to the Z line. Desmin is the one which binds the Z line to the sarcolemma, right? I think we all know sarcolemma, skeletal muscle fiber membrane. Actin and myosin you very well know. Actin is filamentous actin. Myosin is, myosin 2 as we call it, which is responsible for, yes, muscle contraction. This is the head of the myosin, right? Neck, hinge and tail. So answer was titin. Moving to question 3, solutions A and B are separated by a membrane. Solutions A and B are separated by a membrane, right? That is permeable to calcium and impermeable to chloride. Solution A contains 10 millimoles of calcium chloride, B contains 1 millimole. Assuming 2.3 RT upon F is 60 millivolt, calcium will be at electrochemical equilibrium when? So you need to find out first the nurse potential of calcium, for which we are going to use the equation 2.3 RT upon ZF log to the base 10 C1 upon C2. Please understand, in the numerator I always take the higher concentration of the ion. In the denominator I always take the lower concentration of the ion, right? Now already I have given you 2.3 RT upon F is 60. So understand now that if this is a representation of my cell, then outside we have 10 millimoles of calcium chloride solution. Inside we have only 1 millimole calcium chloride. But the membrane is permeable only to calcium. So I will only look for calcium, right? So if I first find out the magnitude using the Nernst equation, answer should be, yes, let's first solve the question. Z, I hope all of you can recall, is nothing but valency. So 60 upon 2 valency of calcium log to the base 10, 10 upon 1. This reduces the answer to 30 millivolt. The problem is answer is given as both plus and minus. For this you will have to recap that as the calcium starts to move in, the positive charge will start developing on the inside, which will start repelling the calcium back. A time will come when the concentration gradient becomes exactly equal and opposite to electrical gradient, movement of calcium stops. So the answer is none but plus 30 millivolt, right? Right, plus 30 millivolt. Let's move to question number 4. The following events occur at an excitatory synapse. Ligand gated channels open in the postsynaptic membrane, synaptic vesicles bind to presynaptic membrane, depolarization occurs, opening of calcium channels. You have to arrange them in the correct sequence, right? So basically what happens is, if you recap for me, this is the A alpha motor neuron, right? So what is, and this is, let's say, the postsynaptic membrane. Over here I would like to call it by the term motor end plate, right? Which is a thickened part of skeletal muscle fiber having a very high concentration of acetylcholine receptors, right? These are acetylcholine receptors. Normally, you know, it is negative inside most of the neurons. So first event naturally has to be depolarization. So this gets depolarized. As soon as it gets depolarized, immediately now these voltage gated calcium channels open. Once the calcium channels open, we have inside this the synaptic vesicles containing acetylcholine. So they will come and bind to this membrane, and immediately now they will cause, yes, release of acetylcholine. This acetylcholine will bind to its receptor and will cause immediately influx of sodium ion. Fine, so let's arrange the events. First is C, then D, then B, and then A. So this was the answer, right? Events at the NMJ are very important question for your exam as well. Then given below is the myogram of a single muscle twitch. What is the tetanizing frequency? Remember, tetanizing frequency is 1 upon contraction period taken in seconds. So over here, how do I calculate the contraction period? Please look very carefully. Latent period is from 0 at the point of stimulus. This is nearly 5. So this can be written as 5, and then contraction period is starting from the 5 millisecond. This is now, let's say again, half of 20 and 30, 25. So this has to be a little more than 25. I can take it 26 or 27. So if I take it around as 27, so 5 to 27 means contraction period is given as CP, which is around 22 milliseconds, right? So if I want to find out the answer, 1 upon 22 upon 1000 second. If you solve this for me, you will easily get on the answer 49 hertz. So the answer was D, right? It's a good question, and it's an AIMS PYQ also. It has been asked in the INICET AIMS exam, right? INICET is the Institutes of National Importance Common Entrance Test. So it's a good question, right? Please don't add latent period in the tetanizing frequency. Tetanizing frequency means within how much frequency I should keep on stimulating the muscle so that it goes into a sustained contraction, right? Sustained contraction, that is tetanus. Which of the following is not the role of ATP in muscle contraction? Again, an interesting question. Please remember, ATP causes dissociation of actin from myosin head. ATP is also required for power stroke. What is power stroke? Flexion of the myosin head from 90 degree to 45 degree, right? It causes sliding of actin over myosin. It causes sliding of actin over myosin, right? Causes muscle relaxation by activating SERCA. Absolutely correct. This is also true. SERCA is an example of primary active pump, right? Primary active. So please understand, and it was an easy question, rigor mortis is seen after death. After death, there is no ATP. So there is no role of ATP in rigor mortis. Rigor mortis is the muscle goes into a state of partial contraction. It starts from head to toe, and as the time period after the death keeps on increasing, there is more and more muscles going to a state of sustained contraction. Erlanger-Gasser classification should be on your tips. So which of the following is a property of C fibers? C fibers, remember, slowest conduction velocity of any nerve fiber type. This actually is the correct answer. C fibers are unmyelinated and having the least diameter. So they are actually having slowest conduction velocity. Largest diameter is of fiber A, so this is wrong. Afferent nerves from Golgi tendon are type 1B fibers according to Lloyd and Hunt, so this is also wrong. P ganglionic autonomic fibers are B, this is also wrong. So the correct answer is only option number A, which is slowest conduction velocity of any nerve fiber type, right? Then let's move to question number 8. A 65-year-old man presented with progressive leg weakness and diagnosis of Lambert-Eaton was made by serial repetitive nerve stimulation test. Which of the following channels are affected in LEMS or Lambert-Eaton myasthenic syndrome? So first of all, understand that Lambert-Eaton syndrome occurs due to autoantibodies against the voltage gated calcium channels which are present on the presynaptic membrane, right? So the answer is voltage gated calcium channels on presynaptic membrane. Also understand, when I do repetitive nerve stimulation, that means with physical activity the weakness improves because the autoantibodies are not blocking the 100% of the calcium channels. With repetitive physical activity, the channels which are not blocked by the autoantibodies, they start causing more influx of calcium and the weakness improves. Although on the other hand, in myasthenia gravis, the autoantibodies are against acetylcholine receptors. So what happens is, with repetitive physical activity or nerve stimulation, the weakness worsens because of the depletion of the acetylcholine or the neurotransmitter. Moving on to the next one, which of the following describes a motor unit? So you have to know this, you have to know the types of motor units, S, FOG, and yes, fast oxidative glycolytic or fast anaerobic glycolytic, which are both type 2 muscle fibers. So you have to know the motor units as well. What is a motor unit? Motor unit is basically a single A alpha motor neuron and all the muscle fiber it supplies. So suppose this is a cross section of the spinal cord, then this is, let's say, the A alpha motor neuron. It is giving these 4 branches, right? So this is my A alpha or lower motor neuron, right? A alpha or the lower motor neuron. This, along with its 4 branches, is supplying 1, 2, 3 and 4 muscle fibers. This structure in together now becomes a motor unit. That means single A alpha, 4 branches and the 4 muscle fibers supplied. This together is known as a motor unit. If I only ask you the number of muscle fibers supplied, please remember this is known as innervation ratio. So innervation ratio of the motor unit, which I have just drawn for you, in this case is number 4. So 1:4, right? Because 4 muscle fibers are being supplied. Given below is the graph of the length tension relationship. Which of the following are true regarding optimum length? So multiple correct options, more than one can be correct, right? So this is the active tension curve, this is the passive. Remember, passive tension is due to the stretching of the muscle. As you stretch the muscle will have a tendency to recoil back. That creates a tension inside the muscle. Active tension is due to the proper actin-myosin overlap, right? So optimum length corresponds to a sarcomere length, absolutely true. There are maximum number of actin-myosin cross bridges at optimum length. This is also true. There is minimum overlap. This is false. Optimum length is also known as resting length. Yes, but only resting length is the term used in the humans. So this is also true. So the correct ones are yes, A, B and D. This is the correct answer. Moving to question 11. Each molecule of troponin C can bind how many calcium ions, right? So please understand, troponin is a calcium binding protein present in the skeletal muscle and the cardiac muscle, and each troponin C binds 4 calcium ions. Very important question, right? Phospholamban in the cardiac muscle cells is which of the following? What is the function being done by phospholamban? Right? What is the function being done by the phospholamban? So please understand, phospholamban is a inhibitory protein which decreases the SERCA activity. So try to recap. SERCA normally causes muscle relaxation, right? So if I am giving or asking you about phospholamban, phospholamban is an inhibitory regulatory protein which is inhibiting the SERCA. So tell me what will happen, more contraction or more relaxation? Phospholamban will naturally result in more muscle contraction. Right? More muscle contraction. Moving to question 13, shown below is the electron microscopic picture of a sarcomere. Which of the following represents the H zone? Remember, how do you approach this question? That is very important. The lighter area is always the I band. So this is the complete I band. So B is the half I band. The darker area is the A band. So this entire one is my A band. In the middle of the darker, we have a lighter area, which I am asking you in this question, which represents the H zone. So H is actually represented by the D. In the middle of the H, we have a line which is M line, and please understand, if this is the M line, then naturally you will all agree with me that this is a Z line. This is the another Z line. So C in this figure is actually the sarcomere, the part of the myofibril between one Z line to the successive Z line, right? B is the half of the I band, and A is the same as the A band, right? Moving to question 14, which of the following statements about the smooth muscle is true? Remember, mechanism of contraction of smooth muscle should again be on your tips. It's a very common question asked in the exams. So, very interesting question. For smooth muscle, you just have to remember that the calcium binding protein is the calmodulin. Calmodulin binds with calcium and forms calmodulin calcium complex. This activates an enzyme known as MLCK, myosin light chain kinase, which basically phosphorylates the regulatory chain of myosin, which is nothing but one of the light chains of myosin present in the neck region. So let's go for the options. Phosphorylation of myosin light chain is required for contraction. Absolutely correct. If you do inhibition of myosin light chain kinase, it will not increase, rather it will decrease the strength of contraction. Inhibition of myosin light chain phosphatase. Remember, myosin light chain phosphatase, which is known as MLCP. This is required for smooth muscle relaxation. So if you inhibit the myosin light chain phosphatase, this will increase the contraction. Stimulation of smooth muscle by nitric oxide, it will increase contraction? No, it will increase relaxation, because remember, independent question, nitric oxide acts via cyclic GMP. It is a very potent vasodilator. It's a very potent vasodilator, right? Very potent vasodilator. So which of the following decreases in length during a muscle contraction? Quite a popular question. And before going for the answer, remember, during muscle contraction, the two Z lines come closer together, width of the I band decreases, H band also decreases or it disappears, M line becomes prominent, and A band remains the same. So the A band, I'll show you one picture also. A band remains same, I band decreases, M line becomes more prominent. So answer is I band. Let's look at a picture now. If you look at this picture, excellent picture to understand what is happening. Above, the muscle is in relaxed state. In the bottom picture, muscle is in the contracted state. What you can see, the two Z lines are coming close together. As you see this, you will realize that the A band remains constant. The A band is the same. But what has happened, because of the sliding inward of the I bands, the I band has reduced in width. Just look at the I. The I band has reduced in width, right? The two Z lines are coming closer together. The H band, which is over here, this is H, it has also decreased. So H and I both shorten, but the M line has become more prominent. M line has become more prominent. That means you can appreciate it better because the adjacent area, which is the H, has reduced, right? But the A band stays the same, right? Same picture has been shown in electron microscopic view on the right side as well. Moving to the question number 16, motor end plate potential at the neuromuscular junction is mainly due to which of the following? So please understand, voltage gated is the wrong answer, because voltage gated is on the area adjacent to the end plate once the neuromuscular transmission starts. Again, this is going to be the wrong one. And please understand, the main answer is sodium influx through the ligand gated sodium channels. This only will produce a depolarizing potential, also known as motor end plate potential, MEPP, right? MEPP, right? Motor end plate potential. Most of the energy, question number 17, for strenuous exercise lasting for more than 5 to 10 seconds but less than 1 to 2 minutes comes from which of the following? Very interesting question. This is about the muscle, sources of muscle energy or metabolism during any exercise activity. So just remember that the ATP which is stored in the muscle is used up in 1 to 2 seconds. Then comes the energy from phosphocreatinine. Phosphocreatinine will be broken down and will liberate creatinine plus phosphate, which will again form ATP. This is used up in 7 to 8 seconds. So we say the creatinine and the ATP stores, which is together known as phosphagen system. On the broad thing, the phosphagen system is used up in 8 to 10 seconds. So any activity which is only lasting for 8 to 10 seconds, such as like a 100-meter dash, a 50-meter run, right, a 20-meter run, a 50-meter swim, that all is around phosphocreatinine or phosphagen system, phosphocreatinine plus stored ATP. After that, any exercise which is for more than 8 to 10 seconds and lasting for around 1 to 1.5 minutes, right? So 1 to 1.5 minutes will be dependent on anaerobic glycolysis, because please understand, it takes time for the oxidation of glucose and the fats and the amino acids to occur and increase the release of ATP. So immediately after 10 seconds, anaerobic glycolysis comes into place. So any exercise such as which lasts for more than 10 seconds to around 1 to 1.5 minutes, something like a 400-meter run, a 200-meter swim, they all are anaerobic glycolysis. And then comes oxidation of glucose, which will be required for any activity which requires more than 1.5 minutes. Like, yes, so many, you can tell me a marathon. A marathon runner mainly depends on aerobic oxidation, right? So answer here has to be yes, anaerobic glycolysis. Fine. Let's move to question number 18. A man falls into a deep sleep with one arm under his head. The arm is paralyzed when he awakens, but it tingles and pain sensation is still intact. The reason for loss of motor function without loss of pain sensation is that in the nerves to his arm, what is happening? Remember, this happened when you were sleeping like this with your arm under the head, and when you wake up you can't move your limb. What has happened is the A fibers are much more susceptible to pressure than C fibers. That is the correct answer. Because there is no loss of pain, remember, as you apply pressure, the thickest fibers are compressed maximum. They have maximum percentage reduction in diameter, resulting in maximum failure of impulse conduction. This is also famously asked as an alternative question, and which you should definitely know: horizontal integration with anatomy, Saturday night, PALC, right? Let's move to question number 19. The diagram below illustrates the single isometric twitch contractions of two types of skeletal muscles A and B in response to depolarizing stimulus. Which of the following best describes the muscle A? So first look at the figure for me. As you look at the figure A, you should be able to understand that in this A, the muscle contracts and immediately relaxes. That means the duration of the contraction is very rapid. But in the B, it starts slowly and remains for a very, very long duration. That means timing is much more. But in the red one, it is very, very fast. Yes, so what can you now think about the answer? So type B has to be over here my red muscle fibers, or also called as broadly type 1, mainly dependent on slow oxidative metabolism, and the A are actually type 2, which are fast muscle fibers depending on anaerobic metabolism. So let's go for the answer now. Which one is A? Adapted for slow and sustained contraction? No. That is B. Composed of smaller muscle fiber? That is B. More mitochondria? That is B. Answer is innervated by large nerve fibers? That is A, right? That is A. At which point on the nerve action potential, question number 20, in the figure shown below, is the membrane closest to the equilibrium potential for sodium? So let's look at the figure first. Appreciate here that the nerve's potential or the equilibrium potential of sodium is plus 61 millivolt. This highest value is generally corresponding to plus 35 or plus 30 as written in various books. But you have to understand that this is the only point which is going to be closest to equilibrium potential of sodium. Because in the points B, C and D, I am going close to minus 70, right? So the only correct answer over here is point A. 21. IPSPs or inhibitory postsynaptic potentials are most likely caused by opening of which of the following types of membrane channels? Remember, IPSP means that I am producing a hyperpolarizing potential, negative. So can you think about the answer? Whenever a channel opens, the ion moves from its higher to lower concentration. Ligand-gated calcium is going to produce EPSP, calcium influx. Ligand-gated sodium, again EPSP, sodium influx. Same for this. The only correct answer is potassium. Whether it is ligand or voltage does not matter. More potassium efflux will produce an IPSP, right? Not EPSP but IPSP. 22. Which of the following characteristics of an axon is most dependent on its diameter? Most dependent on its diameter. So please understand, RMP has nothing to do with the diameter. Refractory period is also a property of the channels of the sodium, types of sodium channels. The absolute refractory period is due to the voltage gated sodium channels which remain in inactivation state. So this is also nothing to do with the diameter. Overshoot of the action potential is again the related to channels. So the only correct answer is conduction velocity. More is the diameter, more is the velocity, right? Moving to question number 23. A 5-year-old boy with weakness affecting the proximal muscles is found to have a mutation of a gene that encodes for protein dystrophin. Genetic alteration in dystrophin leads to progressive muscle weakness because dystrophin provides structural support to the sarcolemma by binding which of the protein? So before I tell you the answer, let's look at this picture. This is the dystrophin-dystroglycan complex. Remember, dystrophin is a protein which is connecting these are the proteins which are present intracellularly. Then these are the proteins which are spanning the membrane, such as sarcospan, as the membrane is suggesting the name sarcospan. This is sarcoglycan group, alpha, beta, gamma, delta. So actin, basically dystrophin protein attaches the F-actin to the beta dystroglycan, which further is attached to alpha dystroglycan, which via laminin is attached to basal lamina. So dystrophin provides a structural support, right? And we have two, muscular dystrophies, which is the Duchenne and the Becker's. Duchenne is due to the absence of dystrophin and Becker's is due to deficiency, right? So important question, the actin to beta dystroglycan, right? Z line to M line, I have already told you. Z line to sarcolemma also I have told you, right? Actin to Z lines, this depends, but if I am talking about the point of attachment, then it could be alpha actin. And if I am talking about the entire length of actin, then it is nebulin. Moving to question 24, in an experimental setup, the conductance of the two ions A and B were plotted superimposed on the action potential recorded from the squid axon as shown below in the figure. Identify the ions A and B. Have a look at the picture. What you need to understand here is this is the membrane potential, this is the time given in milliseconds. So what you have to know is that the voltage gated sodium channels are fast to open, fast to close. So please look at this. They are opening quickly and closing quickly. But the B is slow to open and much slower to close. So can I have my answer? A is sodium, B is potassium. Moving to 25, distance from between one stimulating electrode to recording electrode is 4.5 centimeter. When the axon is stimulated, the latent period is 1.5 millisecond. What is the conduction velocity of the axon? Please understand, suppose this is the point. Now you are applying this is the point of stimulus. This is the point from where the response starts, right? Response begins. That means, let us say it is like this, right? So what I want to know is, this is given to me is the latent period, right? Latent period. So if I have given you the latent period, I have given you the distance between stimulating and the recording electrode. Response begins is being recorded with the help of recording electrode. Can I have my answer? Very, very simple mathematics. Very simple mathematics. Yes. Recording electrode, distance between stimulating and recording electrode. How much is it? 4.5 centimeter. What is the speed? Speed is equal to distance upon time. What should be the answer? 4.5 into 10 raised to the power minus 2 meters. Why am I doing meter? Because the answer is given in meter per second, right? And this we are doing a division by 1.5 into 10 raised to the power minus 3 seconds. Now please solve this for me. Immediately you will get the answer. This has to be 30 meter per second. So the correct answer is 30 meter per second. Fine. So I hope at least you people should have got 18 to 20 correct out of this, right? And anybody who is scoring more than 20, that's wonderful, right? So thank you so much for patient listening. Until I meet you next time, take care. God bless you all.